So now we're going to start the conversation on your special senses. Your special senses are the 5 additional senses we have in our body, minus your nociceptors and your pressure receptors and everything in your skin and hair, not your hair, either way. And these include taste, smell, sight, touch, which we've already talked about in the integumentary system, and hearing. So this PowerPoint part is going to cover taste, smell, and the beginnings of eyesight. Of course, eyes are going to be our most complex sense organ. When we talk about our chemical senses, we are talking about anything that has an odorant or whatever that has to be dissolved into mucus for us to recognize it. So remember that we are aquatic organisms. We are all internally aquatic. We are wet on the inside, 0.95% NaCl aqueous solution. And that includes our saliva in our mouth and the mucous membranes of our nose. So our chemical senses involve gustation, which is taste, and olfaction, which is smell. Also letting you know that these are our more ancient sense organs. These were created in an aqueous environment. That's why we still need an aqueous environment to be able to use them. The chemoreceptors respond to chemicals in an aqueous solution. So what we are tasting, what we are smelling is a dissolved chemical. Taste is substances dissolved in saliva, and smell is substances dissolved in the fluid of, and it's mucus, in the mucus of the nasal membranes. Okay, so a taste or a smell is a chemical that you have inhaled or put in your mouth that is dissolved in the aqueous environment of your nose or your mouth, and then your chemical receptors can respond to it. Taste buds are on your tongue. Those 10,000 or so taste buds, most of them are found on the tongue. They are found in papillae on the tongue. Now, papillae is a medical term that translates into nipple-like. And what a papillae is a small bump or projection. So the papillae of the tongue are small bumps or projections that include the taste buds. There's 3 types: filiform, fungiform, and circumvallate. We're not going to worry about filiform because fungiform and circumvallate papillae contain the taste buds. And so here is a diagram of your taste receptors. You can see that the circumvallate or circumvallate are arranged at the base of the tongue, and the fungiform are actually scattered all throughout the tongue. Right behind or posterior to the circumvallate papillae is your lingual tonsils, your palatine tonsils, and then your epiglottis to help keep substances going down your airway when you swallow. If you look at a circumvallate papillae, let me get a brighter color here, you see that the taste receptors are actually down in that papillae, that projection. So not only do the odorants and chemicals have to dissolve in the saliva, but they also have to be able to travel down to where the taste receptors are. And we call those taste receptors a gustatory receptor cell. I need a color that you're actually going to see. Gustatory receptor cell is what we call it. And it looks like balloon, I guess, where your taste pore, so this is to the top of the tongue here. Your taste pore is an opening in the tongue, and your dissolved chemicals travel down to what we call gustatory hairs. These cause a chemical reaction in the gustatory hairs that are transferred to an an electrical reaction with these fibers of the cranial nerve, and those cause an excitation of the cranial nerve to go trigger the part of the brain that responds to taste. As you can see, the tongue is made up of stratified squamous epithelium because it's full it's subjected to abrasions all day long. And here is a slide, a stained slide under about 40x, I would say. No, that's got to be 100x, showing you the gustatory cell, the taste pore opening at the surface of the tongue, and your basal cells with the connective tissue here. It's a very good diagram. So those gourd-shaped taste buds have supporting cells to insulate the receptor, basal cells, which are of course dynamic stem cells, so they're constantly reproducing the gustatory cells or the taste cells themselves. Just like we had basal cells in the integumentary system that is constantly replenishing the skin layers, the basal cells of the taste buds constantly replace the gustatory cells, because once again, the tongue is subject to all kinds of abrasions all day long. There's 5 taste sensations. There's sweet, which is what we are addicted to evolutionarily because sugars are quick energy. And back in, Holocene times, we're in the Holocene times, the sugars were fruits. And so we are addicted to sweet sugars because it means fast energy. Nowadays, sweet means processed sugar, which is not a good thing. We have salt, which is a metal ion, Na plus, not a writer. Sour, which are hydrogen ions. You know that the more hydrogen you have, the more sour something is. So lemons are super sour, but Coke we see as sweet, but Coke is actually on the 2 pH, it's a 2 pH on the pH scale, and so we should be recognizing it as sour, but it has way too much sugar added to it. Bitter, alkaloids such as quinine and nicotine. Bitter was created as possibly the first evolutionary sense in taste to stop us from eating poisons. And then the 5th taste sensation has one that science has agreed upon is a taste sensation. It's umami. And so that is savory. That's like the taste of meat or the taste of okay, really meat is all I can think about, but it's that earthy, meaty flavor that foods sometimes have that you just like think is savory. So another word for umami is savory. In order to taste, not only do your taste buds have to work, the gustatory cells have to work, and the nerve, cranial nerve has to trigger. But first, all that has to happen. We have to have it dissolved in saliva, we've talked about quite extensively, and we must contact the gustatory hairs. All this has to be a thing. So you have to have a wet mouth to begin with. So all those people who suffer from dry mouth symptoms already have limited taste abilities. After we bind the food chemical to the gustatory hairs, we have to depolarize the taste cell membrane to depolarize an action potential in the cranial nerve. Once that happens, okay, so we can decide what we're tasting by what chemical, that is dissolved in our saliva. So sodium is salt, hydrogen is sour, and gustation is in sweet and bitter taste. So that's a little aside. Once we're able to depolarize the cranial nerves, 7 and 9, they carry impulses from taste buds to the solitary nucleus of the medulla. Ooh, this is also a way for you to understand that this is an ancient, oh my God, I'm so sorry. This is an ancient creation because it goes to the medulla first, and the medulla is part of our ancient reptilian brain. These impulses then travel to the thalamus, and from there to the gustatory cortex and the hypothalamus and the limbic system. So our limbic system is like our pleasure system, and we can appreciate good foods tasting good in our limbic system. So here's that pathway. Your taste buds have recognized a chemical. They've depolarized into the cranial fibers that have then sent an action potential up the glossopharyngeal nerve and facial nerve 7. These hit the medulla first. It's an ancient sensation. But then, evolutionarily, we've created longer sensation trains to the gustatory complex and the thalamic center so that we can appreciate and understand what we are tasting. Taste, however, is not just taste. It is 80% smell. That seems like a bit. I've kind of heard 70 before, but still, we'll go with what the slides say. It's 80% smell. You also have to understand that taste is dependent on your thermoreceptors. Is it hot or cold? Your mechanoreceptors tell you if it's mushy, if it's tough, if it's chewy, if it's any kind of chewing sensation. And nociceptors, is it spicy? Is it so hot it's painful? Did you bite your tongue, while you ate it? Those also influence your tastes. And temperature and texture enhance or detract from taste. We know that we can burn our mouths, and we know that things can be so cold that they don't transmit the chemical signal properly. And also, I don't know about you, I am not a texture girl. Like, textures don't put me off. My favorite texture is dry and crunchy. I know that that is weird to some of you, but like, my husband is a texture person, and if it doesn't have the right texture, he can't eat it, he says, which is very frustrating. And I'm sure you all know texture people in your lives as well. Since taste is 80% smell, then we're going to talk about smell next. That also should let you know that even though taste is possibly our most ancient sensory organ, it is not very effective. And like I said, taste really evolved in order to stop us from eating poisons. And then we expanded upon it as through evolution, we began to eat more and more distinctive things. Smell then came along, and smell is a very heightened sense organ, still aquatic. We still need to dissolve the chemical odorants in okay, well, none that says that, but in the mucosa of the nose in order for the olfactory epithelium to be able to sense it. Now, we haven't really talked about the nose yet, so let me get you a picture of the nose. Okay, so here's your nose, if you cut away along the mid-sagittal plane, and we have inferior nasal conchae down here, and we have superior nasal conchae up here. And you can see that the olfactory epithelium projects down to the top of the nasal superior conchae. Now, what the conchae are supposed to do is, as you inhale air, those cause turbulence of the air in your nose, and it helps warm up the air, and it helps get it in touch with the mucous membranes to filter the air. So you actually have air turbulence as you breathe in due to these nasal conchae. So I can't go back apparently in this presentation. The olfactory epithelium extends down through the cribriform plate of the ethmoid bone. That is very important. Oh, here it is. Through the cribriform plate of the ethmoid bone, your olfactory epithelium falls down to the top of that nasal conchae. So the superior nasal conchae is here. As that air is turbulating around, which is not a word, but is being subject to turbulence from the nasal conchae, you see we have olfactory cilia lining here along the mucus. So as the air contacts the mucus, any chemical odorants in the air, and the best example I can give you of a chemical odorant that we're all going to recognize is the smell of a skunk, right? So that is a very strong chemical oil. It's an oil in the air, and the reason it's an oil, and we know it's an oil, it's because once it's sprayed on your skin, it's very, very hard to remove because oils adhere so well. So if that oil gets dissolved into the mucous membranes, it goes and triggers this olfactory cilia. And I want you to see here that we have nervous tissue connect it to nervous tissue. So here we have a bipolar membrane, a bipolar nerve cell. It has two poles here, and then a mitral nerve cell to go to the olfactory tract. But we have the dendrites that are triggered will send an action potential to the nucleus of the olfactory cell, and then it will also send an action potential up to the mitral cell here. Let me circle this. Along with all that, you have supporting tissues and basal cells and lamina propria, which is a supporting connective tissue. Okay. Physiology, the odor-causing chemicals are going to bind to a ligand. And this is going to trigger what we call a G protein cascade, which you're not really familiar with, and you should learn more about it in okay, I don't remember where you learned about it. Maybe it is supposed to be me. But you probably heard of the cyclic AMP cascade. That's usually triggered by a G protein. And there's a picture coming up so I can show you. So the G protein cascade is triggered, which means you trigger a protein that triggers a protein that triggers a protein, which finally takes cyclic AMP. So we go from ADP, that's a D, P, and we go and we take off a phosphate and we get cyclic AMP. And the reason it's cyclic is we just go back and forth between ADP and AMP. But that's cyclic adenosine monophosphate as a second messenger. The cyclic adenosine monophosphate opens sodium and calcium channels, causing depolarization of the receptor membrane that then triggers an action potential, just like in any nerve system. So the olfactory receptor cells synapse with the mitral cells. So remember, the olfactory receptor cells are bipolar. They have their nucleus, and then they have an axon here and an axon here, and they both end in dendrites. It's a bipolar cell. And then the mitral cells process those odor signals. That's their job. And it sends impulses to the olfactory cortex, the hypothalamus, the amygdala, and the limbic system. So the limbic system is pleasure, or not. I don't think anybody enjoys smelling skunk. The amygdala is memories. That's where your memories are housed. So sense trigger memories. So here is that cyclic GMP cascade. This is a G protein. And here it's only showing that it triggers one other protein. Usually there's a lot longer. And here is our ADP. I'm sorry, I'm not good at writing on this. That goes to the cyclic AMP. And that cyclic AMP then comes up and triggers a channel protein, in this case, to let Na plus or calcium 2 plus, which are metal ions, into the cell to cause depolarization to the olfactory cortex. Oh, there is the words. Thank you. Okay, so yes, there we go. Okay, woo, got it all. But okay, so the odorant binding protein is inactive. An odorant chemical comes and binds. The G protein activates and goes, well, it says ATP, that's fine, to cyclic AMP that comes and triggers a channel protein that causes sodium to cause depolarization for the receptor cells. And that is gustation and olfaction, or taste and smell. Our most complex sensory structure is the eye and all of its associated structures. So 70% of all sensory receptors are in the eye. The eye is very evolutionarily complex. It started super rudimentary, and then through evolution became a very, very, very integral part of higher organisms. Most of the eye is protected by a cushion of fat and the bony orbit. I don't care how skinny you are, you have fat in your body. I've said this before. You have to have fat to cushion your organs. You have to have fat for some of your glandular tissues. You have to have fat for your nervous system to create those myelin sheaths. And this is an example of cushioning an organ. So our eyeballs are soft and squishy, right? but we need them to not be soft and squishy, and so we have bones to protect them as well. Accessory structures, because eye and its associated structures, somehow I got off the pin. Is this the pin? Okay. Are the eyebrows, that was supposed to be a check mark, the eyelids, the conjunctiva, ooh, lacrimal apparatus, which is how we produce tears, and the extrinsic eye muscles, and there's a lot of them. Eyebrows are the remnants of having full body hair on our face. Remember, we come from the great apes. They are completely covered in hair. We actually have the same number of pores in our skin as gorillas have on their skin. Our hair is just a lot finer than theirs is. But through evolution, when we started wearing clothes and no longer needed full body hair for warmth, we did retain the eyebrows. Their function is to shade the eye and to prevent perspiration from reaching the eye, as well as any other falling debris. They're supposed to protect us from things falling from our head space and reaching the eye. Same thing with the eyelashes. That is why they still exist on our eyes, is to filter the air around the eye to try to stop irritants from hitting the eye. Your orbicularis muscle, depresses the eyebrows. So pretend like you're frowning really, really hard and depress those eyebrows. And then the corrugator muscles are what move your eyebrows up and in. So look like you're puzzled and move your eyebrows in. The eyelids, fun fact, the eyelids, let me get to the thing I was reading. Let me try to not knock over my water. You don't know what I'm doing, but that's fine. Oh no. Where is it? It was really fascinating. Oh, here it is. The eyelids at 8 weeks, the eyelids start to form. So in the creation of your eyeballs, there's a lot going on. But when you are 8 weeks in utero is when our eyelids start to form. And I wish I could read this whole paragraph thing to you, but it is quite long and I don't want to waste your time. The eyelids protect the eye anteriorly, so to the front. The palpebral fissure is what separates our eyelids and allows them to open fully. And canthi are those medial and lateral angles, which ladies, you love to really project with your I can do this. I can talk. Mascara, not mascara. It is eyeliner. There it is. Eyeliner. The eyelids also have the lacrimal caruncle, which is that part that can get irritated at the far medial section of your eye right next to the nose. It has an oily secretion that's called Sandman's eye. If you ever wake up and you wipe the sleep out of your eyes, that's what is producing that. And then you don't need to know this, but we have connective tissue supporting the eyelids internally on tarsal plates, and your levator palpebrae superioris gives the upper eyelid motility. Eyelids have eyelashes that protect the free margin of each eyelid, and they initiate the reflex blinking. You usually don't have control of your blinking, or you're not paying attention to the fact that you're blinking. We blink because in evolution we lost that third eyelid that would come across our eye and keep our eyes moist. So we blink to keep our eyes moistened. We blink when we're thinking. We blink when we're lying. That's a big tell when you're lying, by the way. But we also just blink out of habit. Right now, because I'm talking about it, I'm blinking excessively. So, but usually you're not thinking about it. When we blink, we have lubricating glands, the meibomian gland and sebaceous glands. You don't have to worry about those. And ciliary glands lie between the hair follicles as well, which we know those exist if you've ever gotten a sty, S-T-Y, in your eye. It's an inflamed ciliary gland or possibly sebaceous gland of the eyelid, and they're very painful and they take a couple of days to go away. Ah, a diagram. So, the eye. Super complex, okay? First, let's talk about some of the tissue. So here's an eyebrow up here. You can barely see the wispy whispers of the eyebrow. And here's your orbicularis oculi muscle that helps depress the eyebrows. Here is that tarsal plate I said supports your eyelid posteriorly. And the palpebral conjunctiva. Conjunctiva, whenever we see conjunctiva, oh, not conjunctiva, sorry, I mean connective tissue. But the palpebral conjunctiva, conjunctiva is a lining on the inside of the eyelids that surrounds the eyeball. So it goes all the way back here and all the way back here, and it can get inflamed. We know it, it's called pink eye. You have glands to keep your eyes moist, to produce oils. You have the cornea. The cornea is the clear window over your eye there that allows light to go through to the pupil in the iris. Once again, eyelashes, eyelashes, eyelashes here. There's another conjunctiva, conjunctival sac here, so you can see the conjunctiva going like that. Oh, okay, so it just does that. I went too far with the conjunctiva before. And then the orbicularis oculi muscle again. Conjunctiva, transparent membrane, lines the eyelids, covers the whites of the eye, and lubricates and protects the eye, and can cause pink eye. The lacrimal apparatus is it's how we secrete tears. Our tears are very important. First off, they contain antibodies and lysozyme as protective mechanisms against stuff coming into our eye. The tears enter the eye superiorly and exit the eye medially. So let me get to the diagram. So superiorly is where the tears are created, up here at the lacrimal gland. And when we produce tears, they come out the lacrimal gland, cross our eye, and gather here, which is where they fall out if you're actually crying, right? But what they're supposed to do is drain from this into the lacrimal sac and drain to your nose, which is also why we have a runny nose when we cry. Extrinsic eye muscles, you have 6, and that is very important for you to know. They enable the eye to move left, right, up, and down. 4 rectus muscles originate from the annular ring. Not important that you know that, but you need to know you have 4 rectus muscles and 2 oblique muscles. So, looking at that, you have 4 rectus muscles. You have medial, superior, inferior, and lateral. Not that hard. And then you have the superior oblique muscle and the inferior oblique muscle. And here's the inferior, medial, lateral, and where's the superior rectus muscle. So 4 of them from the annular ring come in all of the 4 parts of the eye, and then the one that's laterally or medially to the inside. And then you have the superior oblique that crosses that way, and you have the inferior oblique that crosses to the front of the eye here. The lateral rectus moves eye laterally, medial moves eye medially, superior elevates the eye like if you're rolling your eyes, inferior depresses the eye as if you're looking down, inferior oblique elevates eyes and turns it laterally, so also if you're rolling your eyes, and superior oblique depresses the eyes and turns it laterally. The eyeball. It's slightly irregular. It's not a perfect sphere. It's hollow, and it's filled with humors so that the hollow structure doesn't collapse on itself, with anterior and posterior poles. So the anterior pole would be the cornea looking out into the world, and the posterior pole is where all the nervous tissue gathers in the back. The wall is composed of 3 tunics: fibrous, vascular, and sensory. The internal cavity is filled with fluids called humors. I already told you that. And the lens separates the internal cavity into anterior and posterior segments. So let's get to a diagram. So here is the lens, okay? So the lens is the deciding factor between, are you in the anterior eyeball here, or are you in the posterior eyeball here? Because depending on where you are, you're talking about 2 very different things. In the interior eyeball, you're getting light filtering through the cornea. It's hitting the pupil and the lens. The lens is either going concave or convex to be able to focus what you're looking on. The pupil is either dilating or constricting depending on how much light there is. The ciliary body is helping all of that movement happen. In the front and the anterior chamber, we have aqueous humor. Oh, that's not even written here. But this is aqueous humor, which is a very thin, watery fluid that light can travel through. And then the lens separates it all. And back here we have vitreous humor, so V for vitreous. And it is a very jelly-like structure to keep the eyeball from collapsing on itself. Now, what you might not know about the eye is that the structure that we call the retina, which is where our rods and cones are and where we get all the light information transferred into pictures for us, that is actually nervous tissue. Your optic nerve comes out at the posterior eyeball and travels along the walls and creates the retina. That's why if your retina gets damaged, it's pretty much permanent damage because nervous tissue is typically very hard to heal. You also have a very tough covering called the sclera at the back of the eye. It's also the whites of the eye. Sclera is the whites of the eye. Human beings are one of the only animals on the planet that have whites of the eye, and we think that it evolved so we can engage in silent communication, like when we're out hunting or when you're trying to determine what somebody is going to do, you can watch their eyes. It's an interesting evolutionary advancement. The fovea centralis is called the center point that's of the eye, but the optic disc is the blind spot where that nervous tissue isn't anywhere at all. So here's your blind spot right here because none of the retina is right there. That's where the nerve is entering the brain. So there's no way to collect any data right there from light. The fibrous tunic is the sclera, the whites of the eye, and that clear cornea. It's all about protecting the eye. And the cornea, of course, lets light into the eye. The choroid region has three regions: choroid, ciliary body, and iris. Because it has three regions, hint, you should know that it has multiple functions. Hint, you might want to know that, hint, for the test, hint, if we're hinting at things. So if the ciliary body is involved, it's helping light transfer from the cornea into the back of the eye. If the iris is involved, it is dilating or constricting that pupil, and it also supplies blood to all the eye tunics. The ciliary body itself is a thickened ring of smooth muscle bundles that helps hold the lens in place. That's very important. Iris is the colored part of the eye. The pupil is the central opening of the iris, so the black part of the eye. The iris and the pupil regulate the amount of light entering the eye. During close vision and bright light, the pupils constrict. With distant vision and dim light, the pupils dilate. Or if we change our emotional state, pupils typically dilate when something is appealing or requires problem-solving skills. So your adrenaline is kicking in. So in order to constrict or dilate, we have to use the sympathetic center of our nervous system because we have no control over it. You can't force your pupils to constrict or dilate. And those cause the muscles to contract and you constrict the pupil. And then sympathetic stimulation causes the pupils to dilate. The retina is a delicate two-layered membrane. And I want you to remember it's nervous tissue membrane. And here I go pretending, okay, you know what? I'm not going to write it. Nervous tissue membrane. The pigment layer is the outer layer that absorbs light and prevents its scattering. The neural layer, okay, good. So it says it. It's a neural membrane. Contains the photoreceptors, the bipolar cells and ganglion cells, and the amacrine and horizontal cells. Guys, there is a ton of nervous tissue happening here. Okay, so let's look at it. Now, what you have to understand is that light is going to come in here. It says pathway of light, but it's going to go all the way, oh, that's a horrible color. It's going to go all the way to the back wall, okay? all the way to the back wall and hit here first at the pigmented layer of the retina. So light goes all the way to the back. And then it triggers the rods and the cone cells. And we're going to talk about what a rod cell does and what a cone cell does. But these are activated based on the colors or the intensity of light. If it's night or if it's day, you're only activating rods. If it's just light, and you activate cones for color. But those are activated. And then they go and trigger the bipolar cells. And the bipolar cells, once again, are cells that have a central nucleus, but then they have two axons. They're bipolar. And so they get triggered. And they go and trigger the ganglion cells. The ganglion cells, you can see all of their tells merge together and exit the eye as the optic nerve. Okay, so light goes back to the very back layer of the retina, and then excitation travels this way towards the optic nerve. So as stated, those ganglion cells are the last to be excited, but they run along the inner surface of the retina, and they leave the eye as the optic nerve. The optic disc is called the blind spot because it lacks photoreceptors, because that's the site where the optic nerve leaves the eye. So here is your optic disc where there is no retina, because that's where everything's leaving the eye. The retina itself has photoreceptors. Rods are for light and are used in peripheral vision. Cones are for bright light and color vision. Rods came first in evolution, and then cones evolved later. They are found in the macula luteae, which is great, but you don't need to know that, and are concentrated in the fovea centralis, which, okay, that's fine too. Blood supply to the retina. Your eyeball is highly vascularized, and the retina itself needs to be vascularized to keep those nerve cells healthy. So two sources. The outer third receives its blood from the choroid, which is that tough layer that forms the sclera and the cornea. And the inner 2/3 is served by the central artery and vein. So I know about the blood supply of the eye because my mom has lost nearly all her vision in her right eye because the blood vessel hemorrhaged in her eye on the night of my rehearsal dinner the day before my wedding. And she kept it all a secret so that nobody would be concerned about her at my wedding, which was super sweet, I suppose, but she lost a lot of vision due to it. And has had to have shots in her eyes and everything to try to regain vision, which sounds like the worst thing in the world to me. And small vessels radiate out from the optic disc and can be seen with an ophthalmoscope. And you can see it when they rupture because